ALS, also sometimes referred to as Lou Gehrig's disease, is the commonly used abbreviation for amyotrophic lateral sclerosis, a neurodegenerative disorder that causes the breakdown of peripheral motor neurons, which is followed by subsequent denervation of nerve target muscles and ultimately results in muscle atrophy of affected areas. ALS is not understood to be a disease whose sole cause or route of origin is genetic, but there are known mutations that can induce the condition. There is no known cure for the condition at this time, and it is always fatal. The majority of human patients diagnosed with ALS die within 3 to 5 years due to a breakdown and failure of the muscles controlling diaphragm movement and the resulting respiratory failure. Because ALS is characterized by muscle atrophy and nerve breakdown, those looking at the progression of this disease will be interested in molecules which seem to be involved in bodily responses to muscle and nerve injuries. One group of such molecules is microRNAs, because recent studies have found that microRNAs are implicated in the body's response to muscle stress. Other previous studies had looked at 320 microRNA molecules to determine which molecules were most strongly upregulated in the muscle of subjects with ALS, and they had found that the microRNA most commonly associated with the disease's onset was one called microRNA 206, often called MiR-206 for short. The purpose of this project was to determine what the role of the molecule MiR-206 was in the pathogenesis of the disease ALS. MicroRNAs were already known to be involved in a number of key cellular processes, such as cell signaling and the regulation of gene expression, and so there were a number of ways MiR-206 might have been connected to the disease. First, to test whether or not it was the damage to the connection between the muscle and nerve associated with ALS that triggered the upregulation of MiR-206, the levels of this molecule were looked at before and after a surgically induced nerve injury that would denervate a number of specific muscles in the legs. MiR-206 was found to be strongly upregulated in each of the muscles affected by the nerve injury, suggesting that the molecule could play a role in the body's innate response to denervation. This study also looked at changes in the expression of MiR-206 with time in wild-type controls and in ALS mice, and confirmed that the expression of MiR-206 increases dramatically in response to the onset of ALS in mouse models with mutations producing the disease. This allowed for three possible explanations. Either MiR-206 was part of a compensatory mechanism against the nerve degeneration associated with ALS, it could somehow be part of the disease's pathology itself, or it could be a correlation that does not actually have an effect on the disease progression itself. To determine which of these potential roles for MiR-206 in the pathology of ALS was correct, two mutant strains of mice were created. One of these two strains of mice simply had the genetic defect that produced ALS, a mutation of the copper-zinc enzyme superoxide dismutase, while the other strain had both the defect producing ALS and an additional mutation that meant it could not produce MiR-206. Of the two strains of ALS mice, the group with only the ALS mutation was seen to live significantly longer than the strain with both the ALS mutation and the knockout of the gene from MiR-206. Mouse strains with only the mutation knocking out MiR-206 but without ALS did not show decreased lifespans, and the diseased strain with the MiR-206 knockout also seemed to show an even more rapid progression of ALS symptoms than the other group without the knockout. These findings strongly suggested that the upregulation of MiR-206 in mice at the onset of ALS is a natural compensatory mechanism to the destruction of motor neurons and damage to the neuromuscular junction, and that this mechanism naturally slows down the progression of degenerative diseases such as ALS. To further confirm the notion that MiR-206 is a natural promoter of the regeneration of nerve connections to muscle, nerve injuries were inflicted on control wild-type mice as well as mice that had a knockout mutation from MiR-206, and the control mice were shown to experience full reinnervation of their muscles in a significantly shorter period than those mice who had the knockout mutation. MiR-206's ability to help facilitate the construction of new neuromuscular junctions probably helps the body delay symptoms of ALS because it helps produce new connections to compensate for the motor neurons that are being destroyed by the disease. This natural compensation mechanism also helps explain the observation that many ALS patients do not begin to show symptoms until a large fraction of their motor neurons have already been destroyed, at which time there are probably simply not enough neurons left for compensating mechanisms to prevail in maintaining the integrity of the neuromuscular junctions. The discovery of MiR-206's role in recovery from denervation suggests potential for ALS and nerve damage therapy based on modulation of this natural recovery mechanism. Seeing as about 31 out of every million people born will be diagnosed with ALS, any potential treatments that extend the lifespan of patients with this disease or allow them to experience a better quality of life are sought after by many. The production and study of mice which overexpress MiR-206 would be one additional element I would have included in this study, to determine to what extent this natural mechanism for restoring connections between nerves and muscles could counter the progression of ALS, and also to explore how such a change would affect the ability of mice to recover from normal nerve injuries. If a deficit of MiR-206 is enough to slow recovery from nerve damage, it may well be possible that induced overexpression of this molecule would have the potential to accelerate the natural timeline for recovery from such damage.